Okay, Assalamualaikum and good morning, everyone. Thank you for joining us again for the next breakfast at UMHealth. Sorry for the slight delay. So to start things off, we will start with the session on electron microscopy for life science applications by Dr. Leo Bayfen. So what we'll do today is that we will have both the panelists to give their quick talk first, and if we have time, we will then have a quick Q&A. So Dr. Leo Bayfen is actually the head of Century Unit, which is a central facility for advanced research imaging, where she'll be talking about electron microscopy for life science applications. So without further delay, Dr. Leo, the floor is yours.
Thank you, Dr. Anwar. Good morning, everyone. Today, I would like to share the electron microscopy for life science applications. So how we can use electron microscopy for life sciences samples. Yeah. So I'm Leo from Century and also from the Department of Molecular Medicine. So sometimes people will ask, where is Century? Actually, Century just opposite of a Medicafam level 3. Yeah, so we provide services such as a transmission electron microscope, field emission scanning electron microscope, and then MRI services. Dr. Azam will explain afterwards about this. And then we also provide services, for example, consultancy, training program that we can design based on group or individually. And we also organize workshop with an industrial partner. So if you're interested to send a sample or have any collaborations, then feel free to email us. Yeah. So what is electron microscope? Yeah. So here shows a comparison between the light microscope and also the electron microscope. Yeah. So electron microscope normally uses the electrons. So resolving power of a microscope not only limited by lenses, but also the wavelength of the light for the imaging. For example, light microscope using the visible light with a wavelength 400 to 700 nanometers. However, electron microscope uses the electron. So electron have a much shorter wavelength, which is 0.017 nanometers at 5 keV. So this can give us a better resolution down to 1 nanometers. So TM, FESM all uses the electron for the imaging purpose. Yep. So the difference between the SEM and also the TEM, SEM is excellent for studying the surface morphology of the sample. So normally you will see the operator use 5 to 30 keV. However, for the TEM, it uses electron beam 80 to 200 keV, higher keV, because it used to study the ultra structure of the samples. So electron beam have to pass through the sample. Yeah. So here shows the example of the comparison images between the SEM and also the TEM images. So you can see these are the images for the mesenchymal stem cells. So for the SEM, only see the outer surface of the stem cell, but using the TEM so that you can see like the mitochondria here, yeah, and then the collagens, the arrow collagens here, the nucleus of the cells, and also the rough endoplasmic reticulum. So you can see the ultrastructure of the sample. So here shows the differences between the SEM and also the TEM. So if you are working with bacterial samples, here shows the SEM images of the bacterial samples, and here shows the TEM images. Yeah. So using the TEM, you can see the ultrastructure, for example, where is the chromosome, cell wall, the plasma membrane. Yeah. So here shows the ultrastructure of the bacterial sample. But if you're interested for the outer surface of the bacterial, how the morphology look like, then you can choose to use the SEM characteristic technique. Yeah. So how we prepare the TEM samples? So for the TEM samples, there are series of preparation that we have to go through. First, the sample have to be fixed using the glutaldehyde, then wash using the buffer solutions, then using the secondary fixative, which is the osmium, then wash again, then follow with a negative staining using the UA, uranium acetate, then follow with a series of a dehydration method, and then embedded in the EPON, the resins, and then embedded using a certain temperature. Yeah, so I will go through one by one. So for the physicians, this is very important step in the preparation of a biological sample under the EM. No matter SEM or the TM, we need the physician's method. Yeah. So it consists of a two major steps. First is a killing, stop the normal life function of the tissues, and then second is a preservation, stabilize the structure of the tissue samples. So we would like to keep the time between the killing and the fissations as minimum as possible. Then we keep the size of the tissues as small as possible to ensure the fixative can penetrate through. And then this can ensure no changes in their shape and size during the sample processing. Yeah. So during the fixative, normally in our lab, we are using the glutaldehyde. Yeah, but some lab also uses the formaldehyde. Yeah, glutaldehyde or formaldehyde, and then osmium tetraoxide is a secondary fixative so that it can oxidize the unsaturated bond of a fatty acid. The reduced heavy metal, the osmium, will add density and give a contrast to the tissues. Yeah, so that we will have a better contrast for the ultrastructure of the samples. Yeah. So after the fissections, then we follow with a negative staining using the uranyl acetate. So negative staining can use a PTA or UA. This is the common negative staining that used in our lab. Actually, there are different kinds of stain that can be used. Yeah. So why we use this negative staining is because we need to give the contrast to the samples. Yeah, so here shows sample preparations comparison between the PTA and UA. I won't go through into detail. If you have a sample, feel free to get the negative staining, also the fixative from us, yeah, from our lab. So after the negative staining, then we will go through the series of the alcohol dehydrations. Yeah. So for the alcohol dehydration, we are using either the ethanol or acetones. So here are the most common used dehydration agents due to their hydrophilic property, so they can exchange with the water inside the samples. Yeah. Then 35% up to 100% of the alcohol. Then we follow with the propylene oxide and with the EPON, so that we will embed the TM sample into the resin sample, as you can see from here. Then we can do the sectioning using the ultra microtome. So the thickness of each section is about 90 nanometers. So that's how it is thin enough for the electron to penetrate through the samples. Yeah. So we can see the ultra structure of the samples. So here show the ultra microtome that is available in our lab, so that the operator will help to section your samples down to 90 nanometers. Yeah. Then this is how we incubate our samples here. Yeah. So here shows the example of some of our study, like my study using, we try to study the effect of the nanoparticles, how they interact with the bacterial cells. Yeah, you can see here are the bright field transmission electron TEM images. You can see the how the nanoparticle damage the nanoparticle cells. And then we even can see the disorganization of the cell membrane and detachment of the cell membrane from the cell wall. Yeah, so TM can help us to understand the mechanism by seeing the images. Yeah. So if we are interested for nanoparticles, we even can see the interatomic nanoparticles. Yeah. What is what is a nanoparticles? Like for example, this is a silver, or it already oxidized to silver oxide. Yeah. So all these also can be images under the TM. So the TM provided with an EDX, we can do the elemental analysis. And then this is the images of a hard dark stem, is a dark field. So we can see how they interact with a bacterial cell. Yeah. So just now I presented about the TM sample preparations, but for the SEM, the sample preparation is a big difference. Yeah, SEM is used to see, for example, this Addis egg, the bacterial sample. Then we even can do the sectioning onto the bacterial cells. Yeah, all these are the SEM images, which are different from the TEM images that you can see the ultra structure of it. So this is just a surface morphology of the samples. Yeah. So for the For the SEM, here are the series of sample preparations. First, maybe I need to dissect profusions of the sample, then cut the right areas, then follow with fixed sections. Using the glutathione, osmium tetroxide for the POX sections, then follow with a dehydration using the series of alcohol with different concentrations, then follow with a HMDS that we immerse the sample in the HMDS that I will discuss afterwards. Yep, and then follow with air drying. Yeah. Another method is still using a HMDS, which is a drying agents. Yeah. We also can use a CPD, which is equipment, the critical point dry machines. Then we dip the sample into acetone and then put it into the CPD. After all this sample preparation, then we bound it onto the cup using the carbon tap onto the sample holder. Then we can put it under the SEM. Yeah. So the sample preparation is very important. It's because as you can see, the bacterial sample, if we just dry it not using the CPD, the critical point dry, or HMDS, like for example, we're using HMDS protocol, you can see the morphology is well-rounded, the cell appear well-rounded, and dividing cells are well exposed. But however, if we just use a distilled water and then dry it at 40 degrees Celsius, you can see that the bacterial cells are flattened and then string, and then the non-removal of the mucus layer also has obstruct the surface structure as well. So the sample preparation is very important to make sure we see what it is, yeah, from the sample itself. And then we cut the sample, facilitate dehydrations. Then with HMDS, which is a drying agent, then we can directly put it into the air drying, vacuum desiccator, then mount it. Yeah, this is a simpler method, but some of the sample that we need to use a critical point dry machines. Yeah, so this is a critical point dry that available in our lab. So first, we need to put the sample in the acetones, then pre-cool it using the liquid carbon dioxide. Yeah, and then we flush out the samples. After that, we remove the sample from the CPD. That's how it's ready for the viewing. For the mounting, it's very important that we need to coat it with the gold platinum so that we will avoid the sample charging. Yeah? So all these are the example of coating materials. Yep, these are the sample marine diethyl fixed in the 4% glutathione, prepare in seawater, then we dry using the air drying protocols. And then here are the housefly, yeah, the micrograph cell sample after the fixed sections. So we can see how the nanoparticle interact with the cell, yeah. And here are the colors of the pomelo. It's quite interesting field dry of the fungus samples and also pollens using the field drying method as well. Yeah, so I think because of due to the limited time, so I need to hand over to Dr. Azlan. Yeah, Anwar, back to you. Thank you.
All right, thank you, Dr. Leo. All right, so